Hello students, in today's lecture we are going to discuss what are the various methods of extraction of crude drug, that is a topic of 5th unit of Pharmacognosy and Phytochemistry BP504T. So as we have already discussed in the previous lecture, what is extraction? So just to recall, extraction is the treatment of the plant or animal tissues with solvent or a mixture of solvents so that the medicinally or the bioactive constituents they get dissolved into the solvent, and most of the inert matter that remains undissolved, that is discarded, and that is known as the mark. So now moving towards the methods of extraction. If you go for classification of the methods of extraction, we can broadly classify the extraction methods into two categories. One is the conventional method or the traditional methods, which we are using since long. And over the period of time, we have developed some modern methods of extraction also. Just to name some modern methods, here I have written this is pressurized fluid extraction, supercritical fluid extraction, microwave assisted extraction, ultrasound assisted extraction. Now all these modern methods are quite efficient and less time consuming, but in this lecture we'll be discussing only about the conventional methods, and the modern methods will be discussed in the coming lectures. So if we classify the conventional or the traditional methods, we have again two broad categories. One is the hot extraction and another one is the cold extraction. Now hot extraction and cold extraction means simply that whether we are heating the solvent or not, right? So if we are heating the solvent, it becomes hot extraction, and if not, then it becomes a cold extraction. Again, we can classify the hot extraction into two categories. One is the hot continuous extraction and another one is the hot non-continuous extraction. So in hot continuous extraction, the solvent is heated throughout the extraction process, but in hot non-continuous extraction, there is no need to give a continuous heating to the solvent. Now, let us discuss them one by one. Coming to the first method, that is maceration. So maceration is a cold type of extraction where the process is conducted at room temperature. This is otherwise known as a steady state extraction. So what happens here, the drug that we use, it should be coarsely powdered, and which is kept inside a vessel, and the vessel must have a stopcock. Now, after putting the drug material into the vessel, sufficient menstruum or the solvent is added and the cork is closed. Now, the powdered material that is present inside, that gets soaked with the solvent till the solvent penetrates into the cellular structures of the drug material. So as we know, once the solvent penetrates the cellular structure, the phytoconstituents which have the affinity towards the solvent based upon the polarity, so they will get dissolved into the solvent. Now this extraction process is continued for 7 days, and after 7 days we need to filter this and collect the filtrate. Now the filtrate, that is the solvent with the extracted phytochemicals. Now many times this process is repeated with a fresh solvent. That means the mark which is collected after filtration, that is again kept for maceration with fresh solvent, and finally the filtrates are added up and volume is made up. Now this method is suitable for thermolabile constituents as we are not applying any heat here. So if we are doing this maceration process with organized or unorganized drugs, so there is a little variation in the process. uh, for an example, if we are taking any organized part, let it be a bark or a root or a shoot, then in that case the menstruum that we are taking, we have to put whole of the menstruum into the drug at once. But on the contrary, if we are going for this process with unorganized drugs like resins and gums, then we have to put only 4/5 of the measured menstruum into the drug, and you have to keep the rest amount for making the volume finally. So that means suppose we are taking 200 ml of menstruum. So if it is organized drug, then you have to pour all the 200 ml into the drug at once, right? But if it is unorganized, then you have to take 4/5. That means only 160 ml of solvent will pour into the drug, and the rest 40 ml you have to keep for the for making the volume at the last. And the second difference is, in case of organized drug, when you macerate the drug, the filtrate is not adjusted to volume, but in case of unorganized drug, we have to adjust the volume finally. So why there's a difference in these two, in organized and unorganized drug, in case of adjustment of volume? So simply, in case of organized drug, a variable amount of liquid is left in the mark, and as it is organized and it's not practically possible to press and collect all the liquid from that mark, and this liquid contains soluble matters. So if we adjust the volume, a weak product would result from defective expression. On the contrary, in case of unorganized drug, the filtrate practically contains all the soluble matter of the drug, and the small amount of solvent that is adhered to the gummy residue that gets washed off when the menstruum passes through the filter. Hence, adjustment to volume leads to uniformity here. That's why there is a little difference in these two processes. Next is the infusion. Infusion method can be a hot extraction method, or it can be a cold extraction method also. If you are taking hot solvent for infusion, so it will be hot infusion. And if you are taking cold solvent, not exactly cold, means we are taking the solvent at room temperature, then it will be cold infusion. So what happens in infusion? The drug material that is soaked, the drug material which is in powder form, that is soaked either in hot or the cold solvent, and the solvent can be water, oil, or alcohol, and we allow the material to remain suspended in the solvent for a very short time. So the mixture is then filtered, and the filtered collected is known as infusion. One thing you have to remember, that the infusions are meant only for immediate consumption. Moving to the next, that is a decoction. So decoction is a method of again it's a method of cold extraction. It's a method of continuous hot extraction, because we have to apply heat continuously throughout the process. So before going to decoction, we have to keep three things in our mind. The first is that the phytoconstituents that we are going to extract, they should be thermostable. The second one is the phytoconstituents that we need to extract, they should be hydrophilic in nature, because the solvent we'll be using here is water. And the third one is that the raw material, that is the crude drug, if they are hard and woody in nature, then only we'll go for this decoction process. Now what happens in decoction? The drug is boiled with water for a stated period of time, that is usually 10 minutes, and after boiling, the liquid is strained. And this process is mainly used for vegetable drugs of hard and woody nature that I've already mentioned, and they should be thermostable, and they should have water soluble constituents. Next one is the percolation. So percolation is the process of a process where liquid slowly passes through a filter. The percolation process is used for the preparation of tincture, and this percolation process is a cold process. I mean, it's a cold extraction method, and this whole process of percolation is completed in three different stages. The first one is imbibition, second is maceration, and third one is percolation. Now what is imbibition? The powdered drug sample, we have to moisten the drug with sufficient quantity of menstruum. We have to take only that much of menstruum that will just moisten the drug, and we have to allow the moistened drug to stand as it is for 4 hours in a closed vessel. Now the second step is maceration. Now the moistened drug that we have already kept for 4 hours, that we have to pack evenly into a percolator. Then after packing the drug into the percolator, we have to keep a filter paper over the percolator, over the drug bed, and then we have to put a layer of washed sand. We do this so that the top layer of the drug is not disturbed. So after putting the washed sand layer, we have to add sufficient quantity of menstruum so that there is a layer above the drug. Now, while pouring this menstruum, we have to keep in mind that the bottom, that there's a tap at the bottom of the percolator, and that should be open, because if there are any occluded gases, they will pass out. So once the gases are removed, liquid will start flowing. So once the liquid starts coming from the outlet of percolator, the outlet is closed. Then the moistened drug is kept in contact with the menstruum inside the percolator for 24 hours. But during this period, the menstruum dissolves the active constituents of the drug and becomes almost saturated with it. The next or the last step is percolation. So in percolation, the downward displacement of the saturated solution occurs. That means we already have the menstruum and the drug inside the percolator, right? So in 24 hours, that menstruum is getting, it has already been saturated with the phytoconstituents. So when we open the outlet, now this a saturated menstruum, it will start uh, coming down through the column of the drug. So if there is any left phytoconstituents, so during this passage of the menstruum through the column of the drug, it will extract the remaining active constituents, and this extract is collected by opening the outlet. So this is the whole process of a percolator. So here is some pictures of the percolator. You can see this is a percolator. This is a conical shape percolator. So I can show you here this portion here. This is the drug bed. The moistened drug is packed here, and over the moistened drug there's a filter paper, and over the filter paper there is a layer of washed sand. And after putting this washed sand, we have to pour the menstruum. This whole portion is the menstruum. Now here is the outlet knob. So while putting this menstruum, we have to keep this knob open so that the occluded gases will get removed from the percolator. And once droplets droplets start coming, we have to close this and leave this for 24 hours. And when we have to collect, that is the last step, that is percolation, we have to open this tap so that this menstruum will flow slowly through this drug, and all the left active constituents, if there is any, so it will get dissolved into the menstruum and will collect the filtrate here, will collect the extract here. So this is the process of percolation. Now coming to the continuous hot percolation process, or we say this Soxhlet extraction. So when active constituents of the drug are not freely soluble in the solvent or difficult to be displaced from the cells of the drug, then it becomes necessary to extract the crude drugs by the action of hot menstruum for considerable length of time. So for this method, we need a round bottom flask, we need a Soxhlet extractor and a condenser. So this round bottom flask is needed to keep the boiling solvent, and in the Soxhlet extractor we used to there's a body, central body in the Soxhlet extractor, where we keep our drug inside a thimble. Now a thimble is made up of high quality cotton fleece, and we can make this thimble with a filter paper also. We have to make a packet where we can put a powdered drug, and we have to keep it inside the Soxhlet extractor. Now the Soxhlet extractor has two tubes. One is a side tube, and this side tube carries the vapors from the solvent in the flask to the condenser, and there is a very a thin siphon tube which siphon over, means it transfers the extract from the Soxhlet extractor to the flask. Finally, there is a condenser in which the vapors of the solvents are condensed again into solvent. We can clearly understand this by this picture. Say this is the round bottom flask which is having solvent, and this flask is kept over a mantle which is the heat source, because it's a hot continuous extraction. So we have to supply the heat continuously, right? Now on the mouth of this round bottom flask, the Soxhlet apparatus is attached. Now this portion where the pointer is, here we have to put the thimble. The thimble is containing the powdered drug material. Now when we heat this solvent gets evaporated, right? So once the solvent gets evaporated, the vapors will move upward, and here is a side tube. So the vapors will escape to the side tube, and from the side tube the vapors will move towards the condenser. Now once they move into the condenser, the vapors will condense into the solvent again, and it will fall directly into the thimble, that is where we are having the powdered drug material. Now slowly it will percolate. The principle is percolation, okay, but here we are applying heat, so this is continuous hot percolation. And in English, the simple meaning of percolation is to pass through. That means the solvent is slowly passing through the drug. Now, when it passes through the drug, it extracts the soluble components which are soluble into the solvent from the drug, and so that the phytoconstituent they passes into the solvent. So when and the solvents are and the extract is collected here, and when it reaches to this limit, this is the siphon tube which transfers this extract back into the round bottom flask, and this process is repeated. So we continue this extraction at least for 48 hours to get an efficient extraction. So this was the method of Soxhlet extraction. So in today's lecture, we have completed all the traditional methods of extraction, and in the subsequent lectures, we'll discuss the modern methods one by one. Thank you.